Hello AP Biology students, we're not going to go over a specific section here in Unit 4, but we're going to review the process of meiosis. Remember that the first part of this unit is going to focus on mitosis in the division of body cells. In the second part of this unit, we're going to be covering meiosis and how this process produces sex cells, those being sperm and egg cells. For humans specifically, you can see we're going to start out with a diploid cell that has 46 chromosomes. During interphase of meiosis 1, the cell is still going to go through replication. You can see this is why I use a darker color to represent that doubling of the DNA. Now, in this case, we're not doubling the amount of chromosomes. We're doubling the amount of DNA, specifically in the form of chromatids. So at this point during meiosis 1, we still only have 46 chromosomes, but each chromosome consists of sister chromatids. During meiosis 1, the cell is going to split and have the number of chromosomes. So each resulting cell will only have 23 chromosomes. This is going to be different than mitosis, where we split the chromosomes up into their individual chromatids. In each of these cells, our chromosomes are still going to have their sister chromatids. During our second round of division, meiosis 2, we are still going to see the cell splitting up the chromosomes, but in this case, they're going to be splitting up the chromatids. The resulting cells are going to be haploid, but still have the 23 chromosomes. Overall, you can see that we're going from a diploid cell with 46 chromosomes to 4 haploid cells that have 23 chromosomes. And this should make sense because, again, sex cells should have half of the number of chromosomes, since the other half are going to come from the other sex cell. Here's another way of looking at it. You can see during G1, we have an amount of DNA that doubles during S phase of interphase. During G2, we're prepping for cell division, and after cell division, we're going to have the amount of DNA. In this case, the DNA is going to be split by the homologous chromosome. So each chromosome in a homologous pair is going to be separated. During division 2 or meiosis 2, we're going to have the chromosomes again. But in this case, the halving occurs where the sister chromatids are going to be split. And after the second round of division, you can see we have half the amount of DNA that we originally have. This again should make sense since we are making sex cells that have half the amount of DNA. Here's another way of looking at it. You can see during meiosis 1, we are splitting up the number of chromosomes. Our original cell has 2 chromosomes, but each cell then after only has 1. It has half the number of chromosomes. You can also see that the cells here have chromosomes that are genetically different. They have different parts of the parental chromosomes, that being from the mother and father. This genetic variation is due to crossing over, where the genetic exchange of DNA occurs between the homologous chromosomes. You can see during meiosis 2, the chromosomes here are going to split up just like they do in mitosis. You can see this chromatid is going to go in this cell, and this chromatid is going to go in this cell. You can see at the end of meiosis 2, each of these cells is going to have a chromosome that's genetically different from each other. These cells are also going to have half the number of chromosomes than the original cell. You can see our original cell had 2 chromosomes, and these cells only have 1. I absolutely love this diagram that depicts meiosis 1 and meiosis 2. You can see during interphase, the chromosomes haven't condensed yet, but in prophase 1, you can see the chromosomes pair up in things known as tetrads. During this phase, the chromosomes are going to go through that crossing over where they exchange genetic material. During metaphase 1, they're going to align in the middle, but in this case, the homologous chromosome tetrads are going to align in the middle, not individual chromosomes. During anaphase 1, the whole chromosome, again, sister chromatids, are going to be pulled apart. This is different than mitosis because in mitosis, the sister chromatids are pulled apart. Now the whole chromosomes are going to be pulled. You can see the cell splitting here in telophase 1 and cytokinesis. At the end of meiosis 1, you can see we have 2 cells that are haploid or have half the number of chromosomes. Meiosis 2 and mitosis are very similar. You can see that the process looks very similar to mitosis. However, at the end of meiosis 2, we create 4 genetically different haploid cells. These haploid cells are going to be sex cells like sperm and egg. And each of these haploid cells is going to be genetically different from each other.